Okay, ready for a deep dive into the world of rice.
Always up for that. What kind of racy adventure are we talking about today?
Well, get this. Imagine a future where we can engineer rice to be basically invincible, withstand floods, fight off diseases, and nourish like billions of people.
Okay, yeah, that's a pretty amazing future. Rice is already such a vital crop. It's what a huge chunk of the world relies on, right?
Right. And with the global population expected to hit 9 billion by 2042, figuring out how to grow more and better, rice is kind of a big deal.
Yeah, no kidding. Feeding that many people is a massive challenge.
It really is. And thankfully, there are some brilliant minds out there working on it. That's actually what today's deep dive is all about. We're taking a close look at this scientific review article, Insertional Mutagenesis Approaches and Their Use in Rice for Functional Genomics.
Wow. Insertional mutagenesis sounds kind of intense.
Yeah, it definitely sounds like something out of a superhero movie, right? But it's really about how scientists are manipulating the DNA of rice to figure out how to make it even better.
Oh, interesting. So, like, changing the DNA on purpose, is that what mutagenesis means?
Exactly. And while that might sound a little scary, mutations actually happen all the time in nature.
Oh, right. I mean, that's how we get different kinds of everything, like all those crazy dog breeds. It's all just changes in their DNA, right?
Exactly. It's how we get different varieties of apples, different colors of roses, you name it.
Okay, that makes sense.
Mm.
So by inducing mutations, scientists can sort of speed up the evolutionary process in rice and see what they get.
You got it. It's like fast forwarding through evolution to find those beneficial traits that could help us grow more resilient and nutritious rice.
And insertional mutagenesis, that's like a specific way of doing this, right?
Exactly. It's like they're inserting a little tracker onto specific genes to see what those genes actually do.
A tracker for genes. Okay, now that's cool. I definitely want to hear more about how that works.
Oh yeah, we're about to get into all of that. Stay with us. So you're saying scientists can track genes? How does that even work?
Well, one way they do it is with this thing called tDNA.
tDNA. What is that? Sounds kind of sci-fi.
It is pretty high-tech stuff. Basically, it's like a little snippet of DNA from bacteria.
Bacteria. So like, are they injecting rice with bacteria?
It's not quite like that scientists have figured out how to manipulate this tDNA, you see. They use it to insert specific genes or genetic markers right into the rice genome, like a little delivery system.
Okay, so it's like a tiny little package they can use to deliver stuff. And what are these genetic markers exactly? Like little flags?
You got it. They're like flags or tags that help researchers easily identify the genes they're interested in.
So they stick this tDNA onto a gene, and then they can track where it goes or what it does.
Exactly. Think of it like a bookmark with a bright ribbon. You stick it on a page in a massive library so you can find it again easily. The tDNA helps scientists find the gene they're looking for in the vast library of the rice genome.
Oh, that's a good analogy. I can totally picture that. So with this tDNA, they can just insert whatever genes they want into the rice?
Well, it's a bit more complicated than that, unfortunately. Remember how we talked about mutations happening naturally? Sometimes this tDNA, it has its own preferences about where it inserts itself.
Interesting. So it's not a completely random insertion.
Right, it's like our tDNA bookmark. It might have a preference for certain types of pages or chapters.
Okay, but if they can't fully control where it goes, how is that helpful?
It's still incredibly valuable because scientists are figuring out those preferences. They're
Mm.
learning more and more about how to target specific regions. It's like learning the layout of the library.
So it's getting more precise over time. That makes sense. But tDNA isn't the only tool in their arsenal, is it? The article also mentions something called transposons, right?
Yeah, transposons, those are really cool.
Those are those jumping genes, right? I remember learning about those in school, but I'll be honest, I don't remember much. What makes them so special?
Well, as their nickname suggests, they have this amazing ability to move around within the genome. They can jump from one location to another.
Wait, seriously, like they have some kind of superpower. They just jump around.
Pretty much. And scientists can actually use this to their advantage. They can insert a transposon into a gene and disrupt its function.
So it's like turning off a gene. And how does that help them figure out what the gene does?
Well, they can observe what happens to the plant when that gene isn't working. And here's the really clever part. They can sometimes get the transposon to jump back out of the gene.
So they can turn it back on.
Exactly. It's like they can flip a switch on and off.
Yeah.
It's a really elegant way to study gene function.
Yeah.
And, it gets even more interesting.
More interesting than jumping genes.
Well, scientists have come up with ways to trap specific types of genes, too.
Trap genes. Okay, now that just sounds like something out of a movie. What do they use? Tiny nets. Not quite tiny nets, but I like how you're thinking. Okay, so gene traps. What are those all about?
So genes have these important parts called exons. They're like the instructions for building proteins. And proteins, well, they do all the heavy lifting in a cell.
Right, proteins are essential. Okay, so what do these gene traps do to exons?
The gene trap is designed to insert itself right into an exon. It disrupts the gene's ability to make a functional protein.
So it's like sabotaging the gene's instructions.
Exactly. And by seeing what happens to the plant when that specific protein isn't made, scientists can figure out what that gene normally does. It's really
Okay,
clever.
it is. But that's not all, right? There were also promoter traps and what were they, enhancer traps?
You got it. It's like having a whole toolbox for controlling genes. Promoter traps, those target the promoter region of a gene that's like the on-off switch.
So they can just turn a gene off, even if it's still there. Why would they want to do that?
It lets them see what happens when a gene is silenced, right? What's different about a rice plant that doesn't express a particular gene? It helps figure out what that gene is normally responsible for.
That's so cool. It's like they're pinpointing the exact function of a gene. And then what about those enhancer traps? Are those similar?
Kinda. They also target the control mechanisms of a gene, but instead of an on-off switch, enhancers are more like volume knobs.
Volume knobs.
Yeah. They control how much a gene is expressed, like is it whispering or shouting its instructions?
Oh, that's a great way to put it. So with these traps, scientists can fine-tune how active a gene is.
Exactly. And often, they'll combine these traps with the tDNA we talked about earlier. Talk about powerful tools.
It's like they're conducting a whole orchestra of genes. This is amazing stuff. But what happens to all this information? They're collecting all this data on individual genes, but how does it all come together?
Well, that's where it gets even cooler. Scientists around the world are working together to create these massive databases of rice mutants.
Wait, really? Like a global rice detective agency?
Exactly. They're pooling their resources, sharing their findings, and building this incredible library of genetic information.
So it's like a central hub for all things rice genetics.
You got it. And this is where that Rice 2020 project comes in.
Oh, right. That was all about figuring out what every single gene in the rice genome does, right?
Exactly. They didn't quite hit their 2020 deadline, but the project played a huge role in creating these databases.
And now researchers everywhere can access these databases.
Yep. Imagine needing to make rice more flood-resistant. You could search the database for mutants that already show that trait. It's like a shortcut to finding the most promising genes.
That's incredible. It's like having a roadmap for rice improvement, all thanks to these global collaborations.
It is pretty amazing. And it's not just about individual genes either. Scientists are using this data to understand how genes interact, those complex networks that control everything from growth to stress response.
So like a deeper understanding of the whole system.
Exactly. It's like going from reading individual notes to appreciating the whole symphony of the rice genome. And speaking of cutting-edge stuff, we can't forget about CRISPR.
Oh, yeah. CRISPR. It's everywhere these days. What makes it so revolutionary for rice engineering?
It's this incredibly precise gene editing tool. It's like having molecular scissors that can cut and paste genetic material with pinpoint accuracy.
So they can just go in and edit the rice's DNA directly.
Pretty much. It opens up some wild possibilities. Imagine engineering rice with enhanced nutrition, disease resistance, all kinds of things.
Wow, it sounds incredibly powerful. But there must be challenges, right? Rice is complicated.
Definitely. One thing is that rice can have multiple copies of the same gene. So even if you edit one, the others might compensate, and you don't see the change you expected.
So it's like a multi-layered puzzle.
Totally. But that's what makes this research so important. We need to understand those complexities to truly unlock the potential of rice.
This has been an incredible deep dive. I've learned so much.
Me too. It's amazing how much progress has been made in rice genetics.
It really is. And it sounds like there's so much more to come.
Absolutely. And for anyone listening who's now craving some delicious rice, I don't blame you.
Huh. Same here. And for those who want to delve even deeper into this fascinating world, we'll have links to all the resources we mentioned in the show notes. So until next time, keep those rice cookers fired up and your curiosity burning bright.